the streets I used to own.
Kenapa memilih ilmu kelautan? Sebenarnya lebih di oceanografinya. Kelihatannya masih langka. Kayaknya sih cukup unik, makanya jadinya tertarik untuk mencoba sesuatu yang baru.
Feel the fear in my enemy's eyes. Listen as the crowd would sing. Now the old king is dead, long live the king. 1 minute I held the key. Next the walls were closed on me, and I discovered.
Waktu zaman saya saja, karena sudah ada Kementerian Kelautan, maka kesempatannya cukup besar. Apalagi sekarang kelautan mulai booming, jadi kesempatannya sangat banyak. Kalau untuk pekerjaan ini yang menyenangkan adalah bisa berkeliling Indonesia. Jangan sampai kekayaan laut kita ini diklaim oleh negara lain. Buat adik-adik yang berencana untuk masuk ilmu kelautan, bidang ini masih sangat membutuhkan kontribusi dari adik-adik sekalian. Semangat aja, tidak sulit dan cukup menantang.
Mada kok itu Mada? Mada. Halo Mas Mada. Halo.
Halo, selamat pagi.
Iya, udah dimulai belum ini? Belum.
Oh, ini Pak, baru mau dimulai, Pak. Baru mau dibuka.
Oke.
Baik.
Ya, Pak. Videonya kok video apa namanya? Tadi ada mantenan kayaknya, kayak ada apa gitu.
Pak. Kita mulai nggak, Pak?
Ya.
Video promosi kelautan, Pak.
Betul, Pak. Video promosi kelautan. Baik. Good morning, ladies and gentlemen. Thank you very much for your attendance and also your participation in our morning webinar. Good morning from Indonesia and good evening, Dr. Ushijima. It's around 9 p.m. in USA, in North Carolina. And it's a pleasure for me and also an honor for me to be the master of ceremony or moderator for today. And then good morning to our dearest participants. I noticed the participants are mostly our students and then several researchers from other universities and also the research institutions. Thank you very much for your time to attend our second international guest lectures related on marine biodiversity, even though maybe this morning it's more related to marine biotechnology, because we have a distinguished keynote speaker who will give us a lecture about the coral holobiont, pathogen versus probiotics. So we will learn a lot about the coral disease and then how to tackle the problems, how to overcome the problems using the holobiont of the corals. Therefore, this class is very interesting personally for me and also for all the students. Therefore, our beloved students, I invite you to open your camera so we can have a better communication. I guess, yeah, do the open camera. And to open this international webinar, I would like to invite Professor Permata Wijayanti as our PIC for the program. To Professor Dia Permata Wijayanti, time and place are yours.
Thank you very much, Pak Mada. Good morning, everybody. Assalamualaikum warahmatullahi wabarakatuh. I do hope that everybody are in a good health and have a strong courage to join the Dr. Black Ushijima lecture this morning. So thank you very much for Dr. Black that you again donate your time to give your knowledge and sharing your knowledge to our students and our colleagues. Thank you very much for all participants and also Bapak Ibu from various departments that joined this lecture this morning. Although maybe the information and sometimes come suddenly, but everybody managed to join this lecture. So I will be happy if everybody can get a strong and good benefit from this morning lecture. And yeah, time are yours. I return to Pak Mada to start this morning lecture. Thank you very much, Pak Mada.
Thank you very much, Prof. Dia Permata Wijayanti. Oh my goodness. I need to stop the video. These participants. Yep. Okay. Well, ladies and gentlemen, for this morning, we have our keynote speaker, Dr. Blake Ushijima. And for today, Dr. Blake Ushijima will give us a lecture related to the coral holobionts. So what is coral holobiont, and then what is their impact to the coral ecosystem and also coral health? What is their benefit exists in the coral ecosystem as well? Everything will be discussed by Dr. Blake Ushijima. Therefore, to give the lecture, Dr. Blake Ushijima, are you here already? Yes, you are here. Can you hear us?
Yes, I can hear you.
Okay, so yeah, time and screen are yours.
All right, let me. All right, everybody see that all right?
Yep, so we can see the slide very clear. That's right.
Perfect. Hello, everyone. My name is Blake Ushijima. I'm an assistant professor at the University of North Carolina, Wilmington. And today I'm going to talk a little about some of the research I've done and my lab's currently working on, looking at primarily the coral holobiont and both the pathogens and the probiotics. Kind of a little background on myself. I got my Ph.D. at the University of Hawaii, doing a lot of work at the Hawaii Institute of Marine Biology. And also did a lot of work in the middle of the Pacific on this tiny little atoll called Palmyra Atoll, studying corals and coral disease. From there, I moved to Oregon State University, where I predominantly worked on shellfish, mostly oysters, and diseases of the actual oyster larvae. You can see here's a tiny little video of some of the modal oyster larvae. And then I moved to the Smithsonian Marine Station in Fort Pierce, Florida, where I started working on another coral disease that's actually I'm going to get into a little bit later on in this talk. And from there, I started at UNC Wilmington in 2020, continuing my research both on coral disease and coral probiotics. But what specifically does my lab study? Well, our research does involve corals. For those who need a little refresher or not really familiar, corals are colonial organisms. And if you take so here's a nice rice coral colony taken in Kaneohe Bay, Hawaii. Take a closer look at these colonies, you see these tiny little, what look like anemones. These are actually polyps because corals are colonial organisms. A lot of the different individual organisms are working together as one. And if you take a closer look, you can definitely see how they look like anemones because they are related. They're all cnidarians. And this entire thing is an actual heterotrophic animal that feeds on little plankton in the water. And this is considered a single polyp. So all of these little polyps are connected together. However, I don't directly study corals. Now, if you look a little closer, you'll notice that, within the coral cells, they're filled, for a lot of species, they're filled with these endosymbiotic dinoflagellates that are actually photosynthetic, that are for many species, are providing majority of the energy to the coral animal. However, I don't particularly directly study those either. You actually have to go a little closer and looking at the coral and actually study the microbes that are associated with coral. So it's part of what's called the coral microbiome, which is a complex mixture of bacteria, archaea, fungi, protists, and viruses. And this is what I'm most interested in. Just a little background on the coral microbiome. In general, there are three general groups part of this microbiome. You have your commensal organisms that are, benefiting from living on their coral host, but not really harming the coral. You have your pathogenic microbes that, are benefiting at the expense of their coral host. And you also have the mutualistic microbes that are benefiting from the coral, but actually provide some kind of benefit to their coral host as well. And this is what my lab is very interested because of this interplay between these mutualistic, commensal, and pathogenic microbes. Because when you have instances of, let's say, your depletion of, or either your mutualistic microbes, or there's some kind of problem with the host immune system, you can actually have overgrowth of these pathogenic microbes, and that can result in disease. One of the earlier diseases I worked on was called acute Montipora white syndrome. So this was one, I did a lot of research in Hawaii. And this is called what's tissue loss disease. So you can see here the normal coral, color of this coral is red, brownish, orange color. However, you see here there's white patch. So you've seen in the news a lot about coral bleaching. This isn't actually coral bleaching, even though it appears white. So for coral bleaching, you actually have a loss of those symbiotic dinoflagellates that are living within the corals. So the coral tissue actually becomes clear because a lot of their color comes from these endosymbionts. However, this is actually a destruction of the coral animal tissue itself. So this is exposing its underlying skeleton. So you can't really recover from that. So if you remove the stressors that cause bleaching, corals can recover from bleaching. Unfortunately, for tissue loss diseases, it results in the actual death of the coral animal. And when conditions are, right for the disease or the pathogen that allows them to, spread much easier, you can have coral outbreaks. So here's kind of an example of what can happen when you have an outbreak of these tissue loss diseases. You can just see the white skeleton. These are all dead corals. And because this area had such high coral cover, it allowed for very easy transmission from colony to colony, basically spreading through this area. This was in December 2011, and basically wiping out hundreds and hundreds of coral colonies. Now, for a lot of these coral diseases, it can be caused by a variety of different things. They could be both environmental or abiotic, biotic factors. For some of these diseases, they are caused by bacterial pathogens. So infectious bacteria that can infect the coral, causing a myriad of different disease signs. And one of these pathogens actually associated with acute Montipora white syndrome is a bacterium called Vibrioliticus. Now, this is actually a fairly widespread bacterium. It's a very interesting bacterium because it infects not only a wide, it's been implicated in disease for a wide number of species of coral in the Pacific. It also has been responsible for a number of acute mortalities associated with shellfish larvae. So there's a variety of different types of shellfish, as well as a range of other organisms that can cause disease. And some of these are not natural hosts. There are more, laboratory experiments testing if they can cause disease. But, as you can see by this list, it's a fairly pathogenic microorganism. Now, in terms of coral, it can sure do a lot of damage, as you've seen by that short video clip. But just give an example. Here's a little fragment of rice coral, Montipora capitata. And this is pre-exposure to Vibrioliticus. And you can see this is what happens. This was only 48 hours later. You can see there's pretty much nothing left. It stripped the entire fragment bare. And for about between 70 and 85% of all the fragments exposed to this particular bacterium, this is what actually occurred. So I also have a short video of what can occur. So this is about 24 hours post-exposure to Vibrioliticus. You can see here the beginning of a tissue loss lesion. And this is kind of a time lapse of what occurs over the next 4 to 8 hours. So this orange part area this is the healthy tissue, and this is where the start of the lesion, and you'll see where the tissue loss progresses. You can see here, it's a very good video of just demonstrating basically the tissue just melting away. At some point, the tissue just starts to die and turn a green color because it's decomposing. And you can see how much the pathogen grows in this particular container. You can see the mucus strands becoming actually visible because they're loaded with pathogenic bacteria. So that was just over a period of 48 hours after the first signs of disease. So you can see how damaging this particular bacterium could be to coral reefs. So one of the interesting studies that we did was we found that when we were looking at different strains of Vibrioliticus, we found a lot of variability between the different strains. Some are more pathogenic than others. That's a completely separate study we can talk about for another hour. But what I really want to focus on here is one of the very interesting observations we made is that there's a link between dysbiosis, or also called dysbacteriosis, or the disruption of the native microflora. So one of the things we found is that when you treat these corals with antibiotics, it perturbs, the mutualistic and mental bacteria. And what that did is it actually allowed for all of these strains to actually have an increased infection rate. So if you disrupt the microbiome that's already present, it actually allows some of these pathogens to have near 100% infection rate. So it's a very good demonstration of how dysbiosis, or how the can affect infection, and really the importance of the coral microbiome in relation to Vibrioliticus infections. But one of the other interesting things that led to is more actually coral, which eventually will lead to my coral probiotics research, is another observation is when we do Coralliticus infections. Remember, I said it's about 70 to 85% infection rate. So not every fragment of coral gets infected when you expose them to this particular pathogen. And one of the interesting thing is, for some of these fragments that survived exposure to the Coralliticus, we found, we cultured the bacteria off them. We found a lot of these bacteria had inhibitory activity, or they seem to produce antibacterial compounds. This is kind of a drop assay plate. So the plate is spread with Vibrioliticus, and you can take isolates from this healthy colony and drop them on the plate. And you can see the zones here that are a little lighter. So we consider that a zone of inhibition, and indicates these bacteria may be producing antibacterial compounds. Now, one of the really great, awesome things we found with these particular bacteria, that they can actually protect against Vibrioliticus infections. So we set up these experiments where we pre-treated some of these corals with these potential probiotics or beneficial bacteria. When we compare them to controls with bacteria that don't produce antibacterials, or just not really doing anything to the coral, we found we can actually protect the coral from infection with this particular pathogen. And for some of them, these are the mortality rates. So these are some of the potential probiotics we actually isolated. Here are our controls. So we can see for the controls, mortality rates are about almost 90%. But for these particular probiotics, just after a treatment with these particular strains, you had a reduction in mortality between 40 and 90%. So it's quite amazing, and demonstrates actually not only the importance of the presence of the coral microbiome and protection against pathogens, but also the fact that some of these constituents of the coral microbiome are actually associated with, can actually may be actively preventing pathogen infection by pathogenic agents. However, this talk is not just on probiotics. We also do a lot of research on pathogens, and the thing is, pathogens themselves aren't defenseless either. You have this evolutionary race between these beneficial microorganisms and the pathogens themselves. If you just study the beneficial microorganisms without studying the pathogens, you're actually never going to win in this particular race, because the pathogens are also continuing to evolve new methods of overcoming these host defenses. And one specific mechanism is called the type 6 secretion system, or the T6SS. I'm just going to abbreviate it here. And this is essentially a molecular needle. So this is thought to have evolved from a bacterial phage. So normally a bacterial phage virus that infects bacteria will land on a bacterial cell and plunges this little needle into the bacterial cell, injecting its nucleic acids. However, the type 6 secretion system seemed to have evolved from this in that, except it's facing outwards, where you have this little molecular needle that can fire outwards from the bacterium. And it'll use that to inject a variety of different potent effector proteins, or actually toxins, into nearby bacteria, killing its competition. And this was actually first discovered in the human pathogen Vibrio cholera. And you can see here is the bacterial cell. And this long line right here, this is actually the type 6 secretion system needle. So you can see how, in comparison to the bacterial cell, how long this system is. And you can imagine that if you had another bacteria that was near here, it can fire this system into the bacterial cell. So it's a very damaging system. Fires, when it once triggered, or fire within about 5 milliseconds for Vibrio cholera, and then instantly and it's on the tip of this needle that's being fired into the competing cell. It's actually decorated with a wide variety of different toxic enzymes that can disrupt a wide range of different cellular functions. Some of them are potentially disrupting protein activity. Some are actually destroying the genomes of competing bacteria. Now, in a somewhat unrelated study, we're working with a few collaborators, Farouk Azam and Dr. Ryan Guillemette, and we were actually working together looking at the type 6 secretion system on Vibrio cholera. And we were looking at Vibrio cholera and how it can, kill various, marine isolates, because it does sometimes live in marine systems, and it needs to compete with other marine bacteria. And, Vibrio cholera can use its type 6 secretion system to actually eliminate a lot of marine bacteria. So for most of the bacteria that Dr. Guillemette tested on, about 90% of the cells that were exposed to Vibrio cholera were killed in these assays. Now, one of the interesting things we found is when you mix Vibrio cholera with Vibrio coriolyticus, you have these killing assays. Actually, the opposite would occur. Vibrio coriolyticus would actually kill 90 to 100% of the Vibrio cholera cells within 4 hours. So it's very quick mortality. So we have this one, invertebrate pathogen basically wiping out this human pathogen. And so one of the even more interesting things, beside the fact that, Vibrio coriolyticus might actually have an active type 6 secretion system, which, besides making it much more scary bacterium, is that when we mutated the type 6 secretion system in Vibrio coriolyticus, as expected, Vibrio cholera is able to kill Vibrio coriolyticus, which was, we actually kind of expected it, it kind of didn't. At first, we thought maybe Vibrio coriolyticus was initially immune to Vibrio cholera's type 6 secretion system, but then we found it wasn't. But then when we deleted the type 6 secretion system in Vibrio cholera and mixed it with coriolyticus that was also mutated, neither of them survived. Both of them survived. Neither of them died. So it was kind of an interesting situation where that, your Vibrio coriolyticus wasn't actually immune to Vibrio cholera, But yet, if when you removed its ability to fire its site secretion system, it was now susceptible to being killed by Vibrio cholera. So kind of the hypothesis we're at now is kind of like a Western standoff where you have Vibrioliticus and Vibrio cholera, but the incredibly scary part is neither of them are immune to each other, but is the fact that the Vibrioliticus system is so potent or so efficient that it can protect itself by basically killing its attacker before the attacker can even do anything. We're still not sure if it's a matter of timing, which would be incredible because for Vibrio cholera, it takes about 5 milliseconds to fire its type 6 secretion system. Or does it just act much faster? But it's quite amazing, but also demonstrates the abilities of this particular pathogen in that they are not defenseless. If you're trying to develop treatments against this particular pathogen, we actually have to take these different attributes in mind. And it's like you have to understand not only your treatment, but the pathogen you're trying to treat. But that's only one aspect of this big story about this particular pathogen. A lot of my years of research on this particular bacterium has found it's a very adaptive and complex organism. Has a lot of different tricks that it can use to not only evade potential probiotics, but also helps it to infect and kill a lot of different invertebrates. This is going to get complicated, but just really to illustrate the complexity of what's going on with this particular bacterium, and that typically you just have it's more than just pathogen colonization causing host damage and disease. Because you have things like the host microflora that's protecting hosts from Vibrioliticus to some degree. However, you have a bunch of mechanisms that are controlled by various environmental signals that can allow this pathogen to inhibit the host microflora or attach to the host tissue. You have a variety of other mechanisms that it can use or virulence factors it can use to damage the host. Some mechanisms are also used for resistance against the host microflora. I didn't even mention the type 3 secretion system, which is believed to directly cause host damage. And there's a whole, a lot of different work that we work on chemotaxis, but that there's very complex microorganisms, but it also has a lot of different virulence factors and mechanisms it can use to, kill the host or evade host defenses. That's kind of some of the things we've been doing with our study on different coral pathogens. However, we're taking what we learned from this research and applying it to other problems and also to develop our probiotics. So one of the other things my lab is working on very intently is something you may have seen in the news, is this coral disease outbreak that's occurring in the Caribbean, especially Florida, which is causing a lot of damage. The remainder of talk is kind of going to focus on that and what's really going on in the Caribbean. And the disease I'm talking about is called stony crow tissue loss disease, or SCTLD. And this is an infectious waterborne disease. The tissue loss disease, you can see here some of the corals affected by it. These regions of exposed just coral skeleton, because it does eventually destroy the coral tissue. And unfortunately for SCTLD, it can cause disease in at least 20 different Caribbean coral species. Has a fairly high mortality rate. For some of the more susceptible species, it can have as high as 100% mortality in some of the areas that it affects. And a lot of the colonies that get infected with SCTLD will die within weeks to months. And this disease also has a very large distribution and it has a very long duration. So it's currently spreading through the Caribbean. I have a few maps on the next slide. However, this disease has been continuously spreading since 2014, which is very concerning. And unfortunately, right now, it's an unknown etiology. We're not exactly sure what's causing this disease. It is an infectious, it is a transmissible disease. Pathogenic bacteria are believed to be involved with progression, but there's no one bacterial agent that is yet to be identified. It's actually one of the things my lab is working on. However, we're going to kind of focus on really tell you about other things right now. Now, if we look at the spread of SCTD over the Florida coral reefs, you can see since 2014, it's slowly spread both north and south along Florida's coral reefs. Just to kind of give you a really brief timeline of what actually happened is, you have the first observations of SCTD in 2014. By 2018, it actually spread from Florida. It was first observed in the greater Caribbean. And unfortunately, this year, one of the coral species, Dendrogaea cylindrus, sorry, my apologies, Dendrogaea cylindrus, was actually declared extinct in Florida. This is one of the more susceptible species, and now you actually can't find it in the wild in Florida anymore. And in addition to this year, has now reached over 100% of the Florida's coral reefs. And to give you an idea of what this particular disease can do, this is Boot Key in the Florida Keys in June 2018. This is about 2 months after SCTLD was first observed. And you can see here these large, massive colonies that are, either if they're white or covered in sediment, they're actually completely dead. So you can see these massive mountain corals just completely wiped out within 2 months of this disease first arriving. So you can see the amount of damage this disease outbreak can cause. And this is kind of a map of its spread throughout the Caribbean. You can see here, 2014, it was first observed in Florida. You can see the years go by, it just spread more and more throughout the Caribbean. Here's an animated map kind of showing you the distribution of different reported outbreaks. It's a very dire situation. We have a very devastating tissue loss disease just basically spreading through the entire Caribbean. A lot of questions. So big question on a lot of people's mind is, how do we stop SCTLD? Well, currently there's a lot of groups in Florida and some places in the Caribbean where they're using actually a proprietary base. It's a paste that's mixed with the broad spectrum antibiotic amoxicillin. You can see here some divers that are applying this antibiotic paste to diseased corals. And depending on the coral species, has between a 67 and 95% efficacy in Florida. Now, it appears to be able to stop, disease lesions on most of the corals it's applied to. However, there are a few major drawbacks. One being, these treatments don't provide lasting protection. You can treat the lesions on the coral. However, this is a waterborne disease. You're not removing the pathogen from the environment. However, what you can do is save some of the larger colonies, allow them to spawn and able to get larval recruits. However, this amoxicillin paste is not, as with all antibiotics, they don't provide lasting protection. You have to keep reapplying it. And, colony level treatments can be detrimental. If you wanted to treat the entire colony with amoxicillin, it actually might be worse. If you remember the mutualistic organisms that are on the coral, if you wipe those out with amoxicillin, that might actually cause additional damages. And antibiotic resistance is a major concern. As with all antibiotics, resistance is a major concern. Microorganisms are continuously evolving and developing antibiotic resistance. Therefore, eventually the efficacy of amoxicillin, as with all other antibiotics, will decrease over time. So when it becomes that point, it's pretty much like, well, what do we do now? So one of the alternatives, as you probably guessed, that my lab's working on is probiotics. So probiotics, as according to the World Health Organization, are live microorganisms, which when administered in adequate amounts confer a health benefit to the host. So when we look back at that coral microbiome chart, probiotics are right over there. There's something that will benefit the host. Now, really, what's the difference between probiotics and antibiotics? Well, for one, probiotic, when we talk about probiotics, in this case, they're bacterial probiotics, or they're microbes that kill pathogenic bacteria, prevent their colonization, while antibiotics are substances that are produced by microorganisms that kill other microbes, or nowadays, there's a lot of them are synthetic antibiotics. One benefit of probiotics is some of them can provide lasting protection. You can have colonization of the host by the probiotics and provide longer lasting protection, while antibiotics must be administered more often because it is just the antibacterial compound. Probiotics can potentially be a preventative treatment, while antibiotics are typically short-lived protection. And another thing to consider is, probiotics can be cheaper to produce. You're growing your treatment up instead of, having to sometimes purchase these expensive antibiotics, which can get a little pricey. When we're talking about probiotics specifically, there's a lot of pros and cons associated with this treatment as well. Speaking of the costs, it can be more economical for the amoxicillin treatments. It costs about over $50 per day for one diver for 1 day of work. However, for the probiotic treatments I'm actually going to talk about later in this talk, it's cost about $6.90 US, so about 1/10 the price of the antibiotic treatments. And, like I mentioned, it could provide lasting protection. And also something that's not really thought about is they can actually provide more complex treatments. Some of these antibacterial compounds produced by these probiotics are beneficial bacteria. Some of them can't be easily extracted. They're produced in very low amounts. They can't be stored very well. They can't be transported, and things like that. And sometimes some of these probiotics would produce a multiple number of these antibacterial, antimicrobial compounds, reducing the risk of pathogens from evolving resistance, because it's much easier for a pathogen to evolve resistance to a single antibiotic than it is to multiple, antimicrobial compounds. I know there are a few cons to using probiotics. There's potential harm to the host, which is one of the reasons why I also promote a very stringent protocol for safety testing, and I'm pretty much understanding everything that occurs, everything that this probiotic can do or potentially can do. Some probiotics may be host specific. This actually might be a good and bad thing. Bad in terms of, you might have to develop a probiotic for each individual host. However, it might be a good thing is that it would mitigate any side effects if it's very specific for, one or a few host organisms. And lots of these probiotics must be culturable and feasible to culture. So this is actually something we're keeping in mind when we're developing any probiotic treatment in our lab, is that, well, if you can't culture it, you can't really work with it. However, if it relies on a very complex and expensive media to grow these particular probiotic, it's not really feasible to use in really any way. For example, the media that we initially used for our probiotics cost about $1 US, and it contains about 3 ingredients. So we're considering all these different aspects of growth as well. Now, the star of our lab is one of our most studied probiotics is called MCH17, and it was isolated from a disease-resistant coral in Florida. Here's MCH17. It's a very nice zone of inhibition, and it produces at least 3 different microbial compounds. And we've already gone through multiple layers of safety checks, checking its safety with healthy corals. We sequenced its entire genome, making sure there's really can't immediately notice any bad things in its genome, and we've understood the chemistry of what it's actually producing. So we know what of the antibacterial compounds it's producing, and combined with our safety testing, we're fairly comfortable with being able to use this on corals. However, the very interesting, something that everybody wants to know is like, how effective is it? We ran a few experiments to see if we can actually treat ongoing infections of stony crow tissue loss disease with MCH17. You can see here is the control corals. You can see by day 8, this entire fragment is dead. There's stuff left, but it's just kind of mushy remnants of tissue. However, the really exciting thing, this is actually an example of a fragment that's from the same colony as this fragment, this control, and about 2/3 or 3/4 of the fragments that were exposed to MCH17 that had disease lesions, about 3/4 of them had disease arrested or slowed significantly. So it's very surprising that it looks like we actually can treat stony crow tissue loss disease lesions directly with MCH17. And for a majority of them, we can maybe stop or slow disease progression. Now, one of the very exciting things about this particular probiotic is it looks like we actually can prevent SCTLD disease transmission, which is very important if you're trying to manage any disease outbreak. So you can see here our control corals. This is a diseased fragment of coral. It has a tissue loss disease right here. And this is actually a healthy fragment of coral from the same species that we've placed in contact with this disease coral. And you can see here, as this disease coral progresses, you have tissue loss start on this particular coral. So for all our control experiments, we had disease progression in the originally diseased coral, but also had about 33% of these healthy corals get stony crow tissue loss disease. This number may seem low, but this is one of the more resistant species because that's all was left in our study site mostly. And also we got some of these fragments from a nursery, and that's with everything dying, that's what you have. However, this is actually consistent with our previous disease transmission experiments. You have about 33% transmission. But the very amazing thing is that for all these healthy corals that are put in contact with disease corals, if we pre-treated them once with MCH17, we actually had no transmission at all. So none of these corals, if you pre-treat them with MCH17, develop disease. Now, even more amazing is that 50% of these disease fragments that are in contact with probiotic treated healthy coral, half of them, the disease actually either slowed or stopped, demonstrating actually the beneficial effects of the probiotic are being transferred from this healthy coral to this disease coral. So it's quite amazing. Not only can we prevent disease transmission, the beneficial effects might be, transmittable between corals. Of course, we've run several controls. We really don't see any negative effects of treatment with this MCH17 at all. So it's very exciting news. However, right now where we are, we have one very promising probiotic. We have a few others that are being studied more. However, we can't really, really don't want to base all our treatments on a single or handful of probiotics. So right now, the major push is to look for more probiotics. So we're focusing on screening healthy and disease-resistant coral colonies. And what we're actually searching for is isolates with antibacterial activity like MCH17, see here is just, again, on their drop plate, you can see these zones of inhibition, demonstrating it does produce antibacterial compounds. However, the main drawback is this is a very low throughput assay. There's nothing wrong about this assay. This is what a lot of people do to test antimicrobial activity. However, it's not very high throughput. And you can see this plate right here. You can test about 20 isolates per plate. And it is very time consuming. You have to make your media, you have to group your cultures, you have to carefully spot all your cultures. And, if we're racing against time to develop treatments for this particular disease, so one of the things my lab is doing actually develop more high throughput isolation and testing technologies for this. So a lot of this work is done by my graduate student, Margaret Walter. And some of the things we're working on is being able to, sample these healthy corals and get their complex microbial community. However, once we get to this stage, using traditional microbiology techniques, it can be very slow, low throughput. You're streaking out plates, you're constantly making thousands of plates, waiting for things to grow, making sure you're trying to purify this complex microbial community, make sure your cultures are pure, and it's very thorough, low throughput. So we're working on adapting technologies used in more biotechnology and pharmaceutical companies to actually increase the throughput of this process. So one of them is through the use of microfluidic single cell sorting. So this is a microcell. You can see the little tiny dots floating around here. So those are microbial cells. And the system uses a targeting algorithm combined with a small piezoelectric actuator. So when you have microbial cells that flow within this targeting range, it's able to dispense them very accurately. So we can take these complex microbial communities and separate them physically without the use of any, special tags or, traditionally trying to plate things out. And using this is combined with a cell sorting system. We actually distribute individual microbial cells in this 96 well, or if we want to use a 384 well plate. So we can actually physically isolate all these bacteria and grow them independently very rapidly. So we can actually fill a 96 well plate, in 5 minutes. In 5 minutes, you'll have 96 individual bacterial cells. So then we can take these different cultures, and we're developing this automated distribution and testing pipeline. We can use our robotic system to actually set up a wide variety of these 96 well plates, whether it be using the systems in combination to test a bunch of different growth medias, or actually setting up a wide variety of different biochemical assays. Now, speaking of the biochemical assays, we weren't too thrilled about the spot plating techniques. So we tried to think of ways of, improving this. So utilizing genetics and microbiology, we started developing higher throughput assays. So just to remind you, it's kind of the older method. You carefully spot out, 20 isolates on this plate, and then the next day or 2 days after, you take a ruler and you try to measure the zone of inhibition to see how much inhibition occurred. This can be time consuming, and it can be sometimes arbitrary. Sometimes you can't really tell how, clear is this zone of inhibition? How big is it? Is there partial inhibition or is there complete inhibition? So we're circumventing this. So we developed these new assays. So those plates you saw with these zones, they're typically spread with a target bacterium you're trying to inhibit. So what we did is we engineered these target bacterium to express yellow fluorescence protein, or YFP. And we modified them in a way that their growth correlates with the amount of expression of YFP. So if you have a target bacterium, let's say growing in a liquid culture, if it's growing normally and it grows up dense, to actually express more YFP, and we can detect that on our special plate reader. However, if you mix it with a probiotic target or a probiotic and your target bacteria, if the probiotic is producing antibacterial and kills the target bacterium, it actually won't express YFP, and we can detect that. And using these assays, we can actually use them in 384 well plates. So completely liquid, we can use our automated system to fill these plates and rapidly screen, 384 isolates per plate. So the machine takes maybe 5-10 minutes to set up one plate. So in substitution of manually setting up and testing 20 isolates per plate, we can rapidly test, set up an assay, incubate it overnight, do a plate reading, which should take a few minutes to read this, so we can basically get our results with 384 isolates, which is greater than 10-fold increase in our throughput. And so we're currently developing this not just with antibacterial activity, but we're also modifying it to use with a variety of other assays as well. We're working with other fluorescence proteins. We're actually doing mixed culture assays. Well, that's our goal, being able to test, does a probiotic work against a mixture of different potential target strains? Because in nature, you're not going to have, one probiotic inhibiting one particular bacterium. So it's kind of some of the stuff we have in our pipeline. Now, what about applications? We talked a lot about screening our probiotics and, developing those libraries, identifying all these things to use against Crow disease. Well, the big question it always comes down to is, what are we going to do? How are we going to apply it in the field? So this is what we're doing a lot with our collaborators at the Smithsonian Marine Station. A lot of help from Dr. Valerie Paul, her chemical ecology lab at Smithsonian Marine Station. Our technician Kelly Pitts, who is very basically organizing all these field trials, and also a lot of different collaborators. And we're actually testing, we're actually the first marine probiotic to be tested on a coral reef. So we're treading new ground here, is that no one's ever done this before. So we actually had to develop technologies to be able to test probiotics on diseased coral. Our first method was kind of our most low-tech method, is where we have these weighted plastic bags, and you just simply pump in the probiotic within the bag to allow for a whole colony treatment. So you can see here's Kelly pumping in probiotics in this bag that's covering a diseased coral colony. Now, for a more direct treatment, we developed this paste that's non-toxic to the probiotic, but we designed it so it can stick to both the coral skeleton and actually the coral tissue. So one of the things we designed about this paste is that normally when you're mixing in your probiotic, it will be more of like a glue or very thick glue consistency. However, once it hits seawater, it activates this polymerization process to then convert from this more paste consistency to it starts actually forming a gel that actually will bind and sit on the coral. However, it's made so it doesn't just sit on the coral for a year. It'll sit on the coral for a few hours, let the probiotics colonize the disease lesion, and then simply come off. And it's made from non-toxic biodegradable compounds. You can see here is a nice image. After the first few seconds of applying this paste, it's somewhat malleable. And then once you leave it, it'll then polymerize into a gel. But it's quite easy to use underwater due to we adjusted its buoyancy, but you can just kind of squirt it on with a syringe and apply probiotics directly to your disease lesion. Now, the field trials are very preliminary, so we really don't have a lot of replicates, but they're very promising. So just with the probiotic paste, we're seeing a lot of these colonies healing, or they're stopping disease spread. And for the probiotic bagging technique, a majority of the colonies that were treated with the probiotic bag were actually healing now, which is very promising. I have to reiterate, it's very low replicate size. more trials need to be done, but from what we can tell right now, the results are very, very promising. I'm very happy with it. That was kind of a side story in terms of coral probiotics. It's not just the corals in the field we have to worry about. There's a lot of efforts, especially in Florida, going on where, you have this very, destructive disease spreading in the wild. So there's massive rescue effort going on where they're taking, removing some of these larger healthy colonies from the wild and moving them to land-based nurseries to actually protect them from stony crow tissue loss disease. This was kind of a pilot study done with Nova Southeastern University with Morgan Short and Joanna Figueredo. And they were having some problems with some of their coral colonies. They were developing disease. So we're actually implementing probiotics there. However, in this case, we're using them as a food supplement. And this is actually kind of correlates with some of the interesting things we found is these corals are a lot smarter than we think, some people think they are, and that they can actually sometimes recognize what's quote unquote good bacteria versus quote unquote bad bacteria. However, this allowed us to actually use some of these probiotics as a food additive because the corals actually don't mind it mixed with their food, and in fact they'll actively uptake food laced with some of our probiotics. So this is a video. This is a coral fed. This is the same genotype of coral. One was fed probiotic feed mixture. This is MCH17, and this is actually Vibrioliticus mixed with food. And you'll see that the coral here, gladly eats up all the food mixed with probiotics. And however, this coral has a very interesting reaction. You can see here, it's basically just, sucking down this food mixture. However, you'll notice on this coral, it'll start to feed, eat the food, but then at this about right now, it actually starts to regurgitate the food. Actually it seems to have recognized, this food is laced with Vibrioliticus and actually throws up the food. You can see this coral is already trying to look for more food. Well, this one is actually regurgitated the food and started to ball it up and produce a lot of mucus, and eventually it'll start to slough it off itself. So it's a very scientific and interesting video of a coral throwing up, but it also demonstrates these corals can actually recognize bacteria. But back to what was actual study is a lot of these some of these corals were having problems where they were developing tissue loss lesions, and we didn't think it was stony crow tissue loss disease. But for a lot of organisms that are kept in captivity, close proximity to each other, and a closed water system, you can sometimes have disease spread. So some of these corals weren't looking too good, and this was February 2021. This is by June after a consistent treatment with the food with with our probiotic. Much happier. There's very nice polyp extension. They're nice and happy. Well, they look happy to me. Here's another coral. This one actually had some disease lesions forming. And this is June right now, or this is June photo taken. It's another angle. You can see how nice this coral looks now. So it's very promising in that not only we might be able to utilize probiotics for these wild corals, but also ones that are kept in captivity. So just kind of recap of our talk, probiotics maybe actually be a viable treatment for tissue loss diseases. As in the lab, at least, it seems to be a very viable treatment. And from what we see with our field trials, it does seem very promising. So we're anxiously waiting to be able to do more trials and see how well that does. Again, it's, people really haven't done this in the ocean. So we're kind of, trying to figure out this as it goes. However, our culturing facility, it will be fully operational very soon. So we're going to continue our efforts on developing and screening more probiotics. We're also investigating more complementary treatments. For example, one of the projects we're hoping to get going is through the use of bacteriophages, or viruses that will infect bacteria. We do have a problem. We potentially have a problem of Vibrioliticus in the Caribbean as well. So combining bacteriophages that can target Vibrioliticus with our probiotics, because bacteriophages are so specific to a particular bacterium, you can actually mix these two treatments together. And of course, we're continuing our investigations with disease, both Vibrioliticus as a species, as well as our efforts on trying to identify what is the etiological agent responsible for SCTLD. And with that, I'd like to thank a whole myriad of different collaborators, especially those at the Smithsonian Marine Station, University of Florida, Nova Southeastern University, Georgia Institute of Technology, and Oregon State University, and a lot of as well as all of our funding sources. And with that, I'd happily take any questions.
Thank you very much, Dr. Blake Ushijima, for your very interesting presentation and also lecture about the probiotics. Personally, Dr. Blake, I'm really mesmerized by the probiotic on coral, because usually we use probiotic to prevent the disease on fish, or even for us, for the human. But for the coral probiotic, well, one of the professor here, and also Prof. Dia Permata, and another professor, Prof. Agus Sabdono, ever did this similar work related to the coral disease and also the probiotic bacteria. We isolated several bacteria at the time, but unfortunately, regarding the lack of fission instruments, we lost all the isolates, unfortunately. But then, this year, maybe next year, we already discussed with Miss Dwi and also Prof. Dia. We will start to work on this again, because coral probiotics is very important in Indonesia, especially because our coral ecosystem is very essential, And also, we already discussed as well, about the possibility of our students to join the research. And just like what we discussed on Monday, maybe our students we can send to your laboratory to do further analysis. Well, ladies and gentlemen, I open Q&A session. If you have any question, please raise your hand, or you also can type your question in the chat box. But before we go to the chat box, I will let anyone who want to ask or discuss directly with Dr. Blake Ushijima, please raise your hand. Okay, the first one is Miss Dwi Haryanti, or Ibu Dudu. Time and screen are yours, Miss Dudu.
Hello, good morning, Dr. Blake.
Morning.
So it's
Morning.
me again. So I'm interested in the tissue loss disease, because I think I saw similar cases here. So what makes me curious is in which depth do you usually find this disease? Do you find it in like a wide range of depth, or only in the shallow, or how?
It depends on where the reef is. They actually don't see it a lot in the fringing reefs, and they don't see it in the very deep reefs. But for as far as we know, it hasn't made it to the Pacific. It hasn't spread everywhere in the Caribbean, so there are some areas that it has yet to spread to. But if you're worried about sodium chloride tissue loss disease in Indonesia, we don't think it's spread to the Caribbean. I mean, the Pacific or Indonesia. But
okay.
Yeah.
Okay, perhaps in some areas in which very close to the, anthropogenic activities, we observe like a lot of diseases there, but specifically in our research area, like apart from that particular area, like I think around 50 miles from there, so further from the anthropogenic activities places, we also did not find. So I was wondering if it's how to say, like anthropogenic related disease, like it occurs because of some activities by people there. So do you also find this kind of how to say, correlation with a lot of activities that is going on, and then you started to find the disease?
For this, well, for this particular disease, there is actually some evidence that they think it's there's a correlation between shipping routes and outbreaks in the Caribbean. In terms of, you know, areas with a lot of people, you know, it's the Florida Reef Tracts. There's a lot of human activity. However, it tends to, when it hits a reef, it's spread throughout the entire reef. There's a lot of things we don't know about SCTLD. One of the things is, I mean, we know it's waterborne, but we don't know if it's the actual pathogen, you know, traveling through the water. Is it attached to, let's say, some kind of vector that's transferring it through the water? We're really not sure at this point. So unfortunately, we really don't know.
Okay. Okay. And in one of your pictures, I saw that the disease looks like there are a lot of sedimentation going on there. Is it correct, or that's just how the disease?
Yeah, so that's a very good observation. So this tissue loss disease has more of a chronic or subacute tissue loss. It was actually not as fast as those Vibrioliticus infections I was showing earlier. So the Vibrioliticus infections have what we're called acute tissue loss. So it's very rapid tissue loss. SCTLD actually doesn't move as nearly as quickly. So it allows time for the skeleton to be colonized by algae and acquire and accumulate sedimentation. You know, it doesn't mean it's less lethal. You know, it is a slower moving disease, but you can end up with like complete colony mortality.
Okay. Okay. Thank you. Thank you, Dr. Blake.
No problem.
Thank you, Ms. Dwi Haryanti, for the questions. And then next, I will give the opportunity to Mr. Evan. Yeah, time is yours, Mr. Evan.
Yeah, thank you. Good morning, Dr. Blake.
Morning.
Thank you so much for your presentation. My eyes are getting wider when you show us that kind of technology to isolate those microorganisms. And you know, in Indonesia, in our lab, actually, we still use the traditional method to isolate the bacteria. So
there's nothing wrong with that. That's what I used to use.
It really times consuming, you know.
Yeah.
For the questions, I have two questions related to the probiotics. First, what do you think about consortium probiotics? And based on your experience, do consortium probiotics give better results rather than single probiotics? And for another question, how do we know that the probiotics could not change its characters when it is applied in the field? I think that's all for my questions. Thank you.
Very good questions. Well, tackle the consortium one, where it actually, I mean, it would depend on the probiotics. So this particular one, MCH17, we actually like, it seems very promising because one, it won't dominate, it doesn't seem to dominate the microbiome. It seems to more integrate itself into the microbiome and then has its beneficial effects. The short answer is every probiotic is going to be different. Some may work better as a consortium. Some may work worse as a consortium. Some may work just as fine as a single probiotic treatment. So we actually have to, unfortunately, can't really give blanket statements like that as like, oh, well, consortium will work better Well, some of these corals might require just one or a few constituents to be added to actually help boost its immune or its defenses, while others might require a consortium. Bacteria are very diverse organisms. They have they're much more diverse than anything else we have, like any eukaryotes. So there's a lot of variety of what can happen. In terms of, do you think the probiotic can change in the environment? Yes, bacteria are constantly evolving. This is actually one of the reasons why we're trying to use probiotics from a specific region in that region. So we're not going to use something alien. Let's say we're not going to take probiotics from Hawaii and start spreading them all over Florida. So we're trying to mitigate that effect because, bacteria do change, but at least, if we take things from Florida and use it in Florida, it's something that's already there, that's already would, be changing with the natural community. There's not much we can do to mitigate that at the moment, but we can take the necessary precautions.
Is it enough, Mr. Evan?
I think I have one more question related to probiotics. Is it crucial for us to recheck the microbiome again after we injected the probiotics in the corals? Is it crucial to determine the microbiome, Dr. Blake?
Yes, I believe so. That's one of the things we're doing now, and it's actually one of the early experiments we did. In the laboratory, we were exposing healthy coral to this MCH17 and looking at, its abundance and trying to make sure, like, is it gonna just dominate the microbiome and become the dominant isolate? Our collaborators at Smithsonian University of Florida are actually looking at the microbiome of the field corals that we're testing and looking how that changes in response to MCH17. And as far as I know, it doesn't seem to, when you douse things on MCH17, it doesn't seem to just completely wipe out everything and take over, which, it might be a protective probiotic, but you really don't want to do that either. So when you're doing trials in the field and future experiments, that is something we're going to keep in mind as well, just to make sure what's going on.
Okay, thank you so much for your answers, Dr. Blake Ushijima. It really helps me a lot. Thank you.
Thank you very much, Mr. Evan, for the questions. Then, is there any other participants who want to directly have a discussion with Dr. Blake Ushijima? Please raise your hand. Do not hesitate. Do not be shy. Don't shy. Is any participant here who want to directly ask? Okay, or well, then we will go to the chat box. Dr. Blake, there are several questions from the audience. The first questions come from Professor Dia Permata. Is the SCTLD reflects the environment around of the reefs? Is there any possibility to track how actually the bacteria enter the reefs from the neighbor environments? What do you think about it?
That's a good question. Yeah, so some of the reefs in Florida weren't in the best shape. Unfortunately, when it spread to the Caribbean, it started affecting very healthy reefs. It also started spreading into a lot of marine sanctuaries that were fairly healthy. So there wasn't. So at first, some individuals thought there was some correlation between higher activities or the health of the reef. But then unfortunately, this disease started spreading to more healthy reefs. And it's actually a terrible realization of, nothing is really immune to this disease. Could it be from the neighboring environments? We have no idea where this disease came from. Like, we don't know why in 2014 the coral started dying. There was a minor bleaching event that occurred prior to the disease first observation of the disease. There are really no strong links to water temperature and the spread of SCTLD. So it's still kind of a mystery.
Okay, Dr. Blake, thank you very much for the answer. And then next, I will, maybe I can give opportunity to Dr. Ali Budi Kusuma. Hello, Mas Ali. I think you can directly ask. Are you here, Mr. Ali? Okay, yes, you are here. Let me spot you. Okay, and then give you opportunity to turn on your audio or mic. Dr. Ali, could you turn on your microphone?
I cannot. Okay. Right.
Okay.
Can you hear my voice now?
Yeah,
yes.
very clear.
Right. Okay. Hi, good morning, Dr. Black.
Morning.
Thank you very much for your presentation. So my name is Ali D. I'm from Sumbawa University of Technology in Department of Biotech. So actually, our university is located in Coral Triangle regions. So
okay.
It is very surprising for me knowing the probiotic technology, because few of my colleagues actually starting to collecting some strains from diseased and healthy corals in order to testing whether they have the activity against some coral pathogens. So actually, my first question was actually already answered from your slide. I'm just wondering how the coral probiotic could be applied in field setting, or that this is presumably would be more applicable under nursery setting, but you said already you put the paste and then you use the like a bag, you trap the things inside. And one other thing that I was wondering is, do you have the chance to test it whether the probiotics are surviving during treatment? So how long the probiotic bacteria actually survive? I'm just wondering that.
That's a good question. They're testing that right now as they're ongoing with the field trials. Unfortunately for the field trials, we have very low replicates, mostly because of COVID is making it difficult to do anything. But in the laboratory, we did also track how long it persists on the coral. We did a few trials. We did in a single dose, and we wanted to see how long we can keep detecting the probiotic. And for at least a month, we can still detect it on the coral. Interestingly, that's when we found it wasn't really dominating the coral microbiome. It would right immediately after inoculation, it would dip like a little below the limit of detection for qPCR. But then sometimes it would, some weeks you can just barely detect it, then a dip below, then a month later you can still kind of detect it. So it's still present. It's not like overtaking everything, which is actually a good thing.
Yeah. So I'm just wondering, are they going to form any biofilm structures, or are they just going to be just distributed evenly on the surface of the corals?
We don't know yet. So one of the things we wanted to work on was trying to do some fluorescence microscopy with it to see kind of where it localizes in the coral, because we really don't know. Yeah. As we don't know if it's just staying on the surface, it's making its way into the gut. We really have not a clue, but we're hopefully we're trying to figure out a good way to do fluorescence microscopy on the corals. If anybody's done fluorescence microscopy on corals, they have a lot of autofluorescence. There's a lot of noise and complications, but we're working on it.
Yeah, sure. Thanks a lot, Dr. Blake. Yeah, thank you. Thank you, Mada, as well, for the chance.
Thank you very much, Dr. Ali, for the questions. And is there any other participants who want to ask directly to Dr. Blake, do not shy. A lot of participants very shy to ask Dr. Blake. Come on, just speak up. Okay, Dr. Blake, and then another questions come from Miss Nilam. What is the best methods to preserve 3 months abundant sample for the marine cross journey from deep ocean to keep the function, identify the microbiome before we observe in the laboratory? Do you have any
to keep
experience?
them live?
Yeah, I guess so, Dr. Blake.
If you can freeze them at ultra low temperatures, minus 80 degrees Celsius, that's with a cryoprotectant protectant like glycerol or DMSO. That's usually, that's how we preserve all our strains, is cryopreservation. If it's, I guess it kind of really depends where you are and what you have available. And I guess she's say for 3 months, or what was the duration?
Yep, yeah, around 3 months. Yeah, preserve 3 months abundance.
Yeah, cryopreservation is your way to go, unless you can somehow freeze dry it, like lyophilization. I mean, that's for long-term storage, and not all bacteria survive that, but that's another method. Some bacteria can do fine for a little while in just plain seawater. It really depends on the bacterium. Yeah, that's a tough one. Yeah.
Well, yeah. Well, okay. It's okay, I guess. Thank you, Dr. Blake. And then another question, well, it's come from me personally, because I'm pretty interested now related to the coral probiotic. Well, Dr. Blake, several articles that I have read, it was written that, for example, bacteria A, it is not pathogenic for coral A, okay? But then in another corals, that bacteria, the bacteria A, has probiotic activity to protect the coral B, right? But then when it goes to another corals, the bacteria A, it will become, what do we say, like pathogenic. It causes several lesions. So how many species of corals that we need to test in case if we have several, okay, for example, we have one prospective bacteria that can be a probiotic. Is there any like standard to test at least like 3 or 4 species?
You mean testing, see if it tests against the targets?
Yeah, well, for example, we have bacteria Pseudomonas, right? And then one article, it mentioned that bacteria have a probiotic potency. And then another article mentioned when they did the isolation for the disease coral, they found Pseudomonas, the same species causing the disease, right? So it means that this bacteria has opportunistic phase in case it will be pathogenic in another bacteria in another coral. So how many coral species that we need to test to make sure our bacteria is not pathogens?
Oh, coral species. I think we tested a little, I think 6 or 8 coral species, some of the common ones in Florida. No, that's a good question. I don't think there's a set amount. I mean, I would, depending on the reef, like if you're long-term studies, you want to eventually try to use something on a reef, I would focus on at least the dominant coral species. Unfortunately, for a probiotic, the safety testing can, be different depending on the strain and what you find about it. But there's a lot of things you look into, in addition to, testing on the corals, highly suggest, sequencing a genome just to it won't tell you if it won't necessarily immediately tell you if it's a pathogen. Sometimes like red flags might pop up if you find that, oh, it encodes genes for this potent toxin or something like that. We're working on a lot of surrogate systems as well. One being like the zooxanthellae, if it's producing toxins that will kill the zooxanthellae or the dinoflagellates that are in the coral, then that's not a good sign either. Those are a little easier and you can grow those in the laboratory, so a little easier to handle than healthy corals. And also the chemistry as well. If the compounds is producing, does it seem to be, or if you can identify them, that's great. Or at least test, like, does it seem to be an antibacterial compound, or does it also kill eukaryotic cells? It doesn't need to be cell lines. You can simply test them on yeast or other eukaryotes like the dinoflagellates, things like that. So there's a lot of things to go about as well, just to really convince yourself.
Because
I think, well, because that's also the thing about bacteria. A name doesn't necessarily mean it's a pathogen or a probiotic. There's a lot of instances where you have something, both Pseudomonas. One can be a coral pathogen, one can be a commensal organism not doing anything, and one can be a probiotic.
Is there any report that you ever read or you ever work with actinobacteria, Dr. Blake? Because as we know that actinobacteria like Streptomyces, they are very promising to produce the antibacterial compounds, right? But is it possible to apply actinobacteria like Streptomyces instance to be the coral probiotics?
Yeah, it's possible. Our collaborating lab, there's this, a few of them are looking at actinomycetes, seeing what compounds they're producing. But it's entirely possible to use them as coral probiotics as well. I mean, they're, like I said, the bacteria are so diverse. I mean, if you look hard enough, you can find something that somewhat matches your criteria. But yeah, I mean, it's a very good idea, because like you said, a lot of them produce antibiotic compounds. But one of the things we also want to make sure is that we're also checking the chemistry, or we're not putting out something that's producing an antibiotic compound used in human medicine or something like that.
Well, because currently I get a grant for actinobacteria associated coral associated actinobacteria to produce antimicrobial, but it's for human pathogens. I never tested to coral pathogens. Therefore, well, we isolated several genus like Micromonospora, and then we also we isolated Streptomyces, and then Escherichia, Gordonia, but those bacteria has antibacterial activity for human pathogens. Because I thought, because the Streptomyces is a filamentous bacteria, so is it possible to apply it as the coral probiotics? But then, because you mentioned to me that another laboratory tried to figure out whether this can be probiotic or not, so I will wait for the report, and then maybe we can also apply in our university, because we have the resources. We are very close to the sea, especially to the Pulau Panjang Islands. It's only around 3 hours from our campus, but we have a marine station, and it only takes 15 minutes to the island. So I think for the students, we have a very big opportunity to work with this. We are waiting for the students who want to join this research group, because every year we have research grant to provide you to do research, and maybe next year we can redirect our focus on coral probiotics. And then, well, next to the other question from Mr. Muhammad Untung. Dr. Blake, how do you think about the role of coral's microbiome in repelling pathogens, since mostly members of coral's microbiome are uncultivated, especially when they are separated from the host coral? or when being moved from their nature habitat, what do you think about the strategy to keep this whole microbiome system function are still well work in lab experiments regarding to find out this function on paralysis?
Okay, I think I understood. Yeah, so there's a lot of things we don't know about the coral microbiome. One of the things is like, well, how much does it change when you bring it into the laboratory? That's a very good point he brings up. There's just more research needs to be done in terms. I mean, the coral microbiome. Yes, there's most of it's unculturable, hard to study, but there's a lot more. Omics studies like metatranscriptomics, metagenomic studies that are being done in the coral microbiome and slowly picking apart of what's really going on. But in terms of unculturable as well, one of the points is, not everything is for every one type of culture media, you're not going to culture everything. But there's also not a lot of research being put on developing culture medias. So it's one of the goals of our lab is we're actually working on developing more culture medias to actually help expand the number of things we can work with. Yeah, I mean, it can't really give a solid answer because we really, comparative to other systems, we really don't know that much about the coral microbiome. Like we really don't even know, why or how, the coral selects for its microbiome, why it's distinct from just the seawater it's surrounding it. Why for some corals they share, a common constituent. There's a lot of work done on like Endozoicomonas. It's a dominant constituent in a lot of corals, but not found in some other corals. We don't know why. So yeah, I mean, I can't. I mean, the short answer is like I don't know. But we're hoping to figure it out.
Yeah, therefore we are the researchers of coral holobionts. Let's work together to answer that mystery, right, Dr. Blake?
Yeah.
Okay, thank you very much, Mr. Muhammad Untung, for the question. And then for our last question, because we have limited time, is from Ms. Nani Koliha. Okay. So, Ms. Nenik said that I have heard that probiotics have side effects. Did the coral probiotic that you have used also have the possibility of producing side effect and such as resistance or the virus or the bacteria be stronger than before? Is it possible to make probiotics that have no effect on the to the environment? Thank you.
I don't think you can completely eliminate the possibility of side effects. I mean, anything is possible. Microbes have been on this planet for millions of years, so they have much more experience than us. But there's a lot of steps we're taking to mitigate it. So one of the reasons why we initially started to really focus on the probiotic we're working on MCH17 is because it produces multiple antimicrobial compounds. Well, like I mentioned before, if you have a probiotic that produces a single antimicrobial, it's much more easier for a pathogen to evolve resistance. However, if it's producing multiple compounds, it's much more difficult to evolve resistance. So that's one of the reasons. And the other reason is the way it kills other bacteria. So it utilizes a lot of the use of the production of reactive oxygen species. So it's can result in DNA damage in target bacteria. So if you damage, or also the denaturing of proteins, or just preventing its ability to function properly by actually damaging proteins in DNA. So that's another reason why we're so interested in it, because you eliminate its ability to grow and actually potentially evolve. You can start to mitigate these issues that can arise. But I don't think there's any way to completely remove any of side effects. We didn't see any side effects on the healthy coral from exposure to this particular probiotic. Is there side effects possible? Yes. I mean, there's side effects possible with anything you use. It's just amount of effort you put into preventing it.
Yeah.
Oh.
Well, thank you, Dr. Blake. Well, basically everything that we are doing in this world has side effect, I guess. Even for example, when we eat something and then our body will produce the dung, and the dung will also contain bacteria, pathogens, and will go to the environment and also will give the side effect to the environment. But maybe the point of the probiotic is how to preserve the corals using the other bacteria. Even though there is another side effect, I think personally, as long as it will not cause any disease to another organisms in the environment, it will be okay, I guess, in my personal opinion. And then, okay, still another question from Miss Dini Riandini. Yeah. So Miss Dini said, my one question is, how do you arrange the actinobacteria function as probiotic but not produce antibiotic substance? So maybe not only actinobacteria, but also other bacteria. Is there possible for us to use certain bacteria even though that bacteria did not produce an antibiotic activity? What do you think, Dr. Blake?
Oh yes, that's a very good question. Like one of the things we're looking into, because the production of an antibiotic is not the only mechanism a probiotic can work. Some probiotics, taken from the human gut research, some probiotics will, sequester nutrients. Some will just physically take up space, competitive inhibition, preventing pathogens from colonizing. There's a lot of other traits that you can look at. For example, if there's trace minerals or trace vitamins that are essential for the pathogen growth, if your probiotic is very good at sequestering them and using them and not providing any for the pathogen, that would actually prevent pathogen growth. That's another mechanism that's also possible.
Okay, thank you very much, Dr. Blake Ushijima, for your time and also your lecture this morning. Well, already evening in USA. Thank you very much. And to close this international guest lecture, I would like to invite Professor Dia Permata Wijayanti. Prof. Dia. Okay, let me give you the spotlight. And yeah, I already unmute you, Professor. Could you? Okay, time and screen are yours.
Thank you very much, Pak Mada. And once again, thank you very much, Dr. Blake Ushijima, for this very wonderful lecture this morning, because I learned many things about how curing, maybe understanding the coral disease that actually is very difficult threat for the coral reef. And I do hope everybody who joined this morning lecture also got a lot of knowledge, new knowledge, maybe, and may help to understand more about how we can cope with the coral disease, because now the coral disease is already become maybe another monster for the coral reefs other than bleaching. But thank you once again. May we can work together to save our corals and in the future, because we need our corals for our next generation. And the corals can give their benefits to our life. And thank you very much for all participants. We will invite you in another opportunity of other webinar lecture. Maybe if you have time, you could join for tomorrow webinar also. Presented by a researcher that work in Jeju Island, Korea. So once again, thank you very much, Dr. Blake. Stay safe and healthy. And yeah, just a small story. I worked with Dr. Kinsey from Hawaii University when I did
Oh,
my student. Yeah. So I met him several times. Even he invited me when I did my PhD, but my professor not allow me to go. So it's very yeah. But I met him many times until the last time I met him in 2000, maybe 12 or something like this in a conference. So yeah, thank you very much and see you someday somewhere. Yeah. Once again, thank you very much, Pak Mada. So I return to you.
Thank you very much, Prof. Dia Permata, for the warm closing remarks. Ladies and gentlemen, before we leave, I would like to invite all of you to turn on your camera. Hence, we can take pictures together. Yeah, please show your handsome and also beautiful face. Do not shy. Let's take picture together. Well, Miss Neni, are you here? Okay, Miss Nene is here. Miss Nene, could you help me to lead the picture session or photo sessions?
Okay, Pak Mada.
Thank you very much, Miss Nene.
Please turn on your camera for first screen. 1, 2, 3. And the second screen. 1, 2, 3. The third screen. 1, 2, 3. The fourth screen. 1, 2, 3. And for the last screen, 1, 2, 3. Enough, Mas Mada. Thank you.
Thank you very much, Miss Nene. And one more time, thank you very much, Dr. Blake Ushijima. We are waiting for our further collaborations. After this, me, Prof. Dia, and also Miss Dudu, we will discuss to prepare proposals, and we will invite you to be part of our research work. And then I hope that everything will be going well for your research in USA. And please do not hesitate if you need any help, just contact me or Miss Dudu or Professor Dia Permata. Thank you very much, Dr. Blake, for the opportunity.
Thank you for inviting me.
Participants. Yeah.
And thank you very much for all participants. 100, more than 100 participants is totally amazing, because last time we had less than 100. So it means that coral disease has a very high researchers who are interested with this. Well, ladies and gentlemen, please don't forget, tomorrow we also have general lectures. The speaker is Dr. Anka from Korea, and she will discuss related to the molecular biology on marine ecosystem for conservation, if I'm not mistaken. So please join us. If you don't get the link or Zoom meetings password, let me know. You can directly email me so I can share to you, or you also can chat me through WhatsApp group. One more time, thank you very much and have a great day. Goodbye. Thank you,
Bye.
Dr. Blake.
Dr. Blake. Thank you Bapak Ibu semuanya, Pak Ali. Yeah.
Terima kasih banyak Bapak Ibu. Thank you very much.
Pak Taufik, terima kasih.
Taufik Nj, ada Ibu Nirwani juga.
Yang dari LIPI, Bu Dini, terima kasih.
Ini saya kepengen meng-unmute semua, tapi kok nggak bisa ya.
Ayo di-unmute dulu Pak Mada.
Iya, coba di raise hand ya Bapak Ibu kalau ingin ini. Karena saya juga bingung bagaimana cara meng-unmute ini.
Nirwani, terima kasih.
Terima kasih banyak. Ya.
Bosnya
Ini Bu Nur Laila sudah saya, ya, beberapa sudah saya invite.
Nur Laila,
Oke, sami-sami. Terima kasih banyak.
Bu Dini,
terima kasih banyak. Untuk materi Bapak Ibu tidak bisa kita bagi, karena ada beberapa hasil yang belum bisa di share sama beliau. Jadi untuk materi we are really sorry. Tapi kalau ingin ditonton kembali silakan Bapak Ibu, karena kita sudah record di YouTube-nya fakultas, jadi bisa langsung aja di YouTube cek saja FPIK Undip Official. Jadi Bapak Ibu bisa menonton kembali untuk materi kuliah pada pagi hari ini. Saya izin permisi Bapak Ibu, karena juga ada rapat lagi.
Banyak.
Terima kasih Prof, terima kasih Bu Dudu, terima kasih Bu Nirwani. Terima kasih Ibu Retno, terima kasih Bu Yeti,
terima kasih.
Pak Budi. Sami-sami Bu Yeti.